Hello everyone, here I'll help you in understanding the concept action potential. It is also called as Naru impulse. How and where Naru impulse action potential is generated, that means where exactly it is produced and how. At the same time, how exactly action potential or Naru impulse is transmitted, that is from one side of the neuron to the other side of the neuron. I have already told you the answer, where exactly action potential is generated. It is generated here. This is the specialized cells which makes our nervous system called as neuron. So this neuron has so many parts. This is the dendroid and this is the cell body, axon, and this is the axon terminal and synaptic knobs. Yes. Okay. So for example, if a stimulus or stimuli is applied here, and a site of the dendrite will collect that stimulus and convert it into some other form of energy, that is electrical current. In other words, we can call it as action potential or nerve impulse. Now nerve impulse which is created here at the site of stimulus, that will be transmitted to the cell body, then into the axon, and ultimately reaches this particular knob, which is actually called a synaptic knob. And from here, another neuron's dendrite will collect that action potential or nerve impulse, and the process will continue. And ultimately, it reaches the central nervous system like brain or spinal cord, where processing of the message takes place. Then regulatory impulses will be released from the CNS to the site of stimulus. Okay, this is how all animals respond to the abiotic conditions. Okay, now let us just understand the structure of the neuron, because all neurons are excitable cells. Why neurons are excitable cells? The simple, because they are always in polarized state. Okay, so this is very important thing to note. Neurons are always in polarized state. What is the meaning of this polarized state? I will help you in understanding this action potential with the simple three steps. The step number one is when the entire neuron is at rest, that is called as polarized state. Next is when the neuron site, a particular site of the neuron is now depolarized. That is nothing but, there is a continuous flow of the ions. Okay, this is nothing but produces action potential. I'll explain it. The last one is once the disturbance has created, that is at a particular side of the neuron, it has to repolarize, that is actually come back to its original state. That is nothing but getting back to this state, polarized state. Okay, so these are the three steps. Let us understand. Step number one, when the neuron is not even conducting any of the action potential or nerve impulse, it is completely at rest. It is at rest. Okay, so now you see the entire neuron membrane. For example, see this one. This is the neuron now, and this is actually the cell body, and you could see the axon here. So I have taken a small portion of the axon and I have drawn that diagram here. So axon is nothing but containing plasma membrane, because neuron is a cell and it also exhibit normal plasma membrane, which all human cells possess. Okay, so here the diagram of the plasma membrane of the neuron looks like this. You could see this is the lipid bilayer, nothing but made up of phospholipids, and this is the lipid polar head, and this one is the lipid hydrophobic tails. Okay, and you could see the lipid bilayer, and between that we all know that all plasma membrane also exhibit the presence of certain proteins, the membrane proteins. They help in the movement of the molecules across the membrane, that is from inside to outside or outside to inside. That is seen. So here also in the neuron plasma membrane you could see that type of proteins here. These are intrinsic proteins, but these intrinsic proteins have a specific function and have a specific name. The blue color ones here depicted as potassium ion channel. So this is potassium ion channel, and this one, the red color ones, you could see around 3 red color proteins, they are sodium ion channels. They are highly selective in nature. They allow only selective molecules from outside to inside or inside to outside. Potassium ion channel actually transport only potassium. You could see the potassium here. Okay, so that is from inside to outside or outside to inside. Okay, whereas sodium ion channels will transport only sodium ions from inside to outside or outside to inside. They are highly selective in nature. Okay, so now in this particular diagram you could see this portion. the outside of the neuron. So here you could see this is the inside of the neuron and this one is the outside of the neuron. Outside of the neuron is filled with a liquid that is actually secreted by the neuron itself. It is called as extracellular fluid. Okay, so here we have extracellular fluid. I just simply write ECF, that is nothing but extracellular fluid, which is present outside the plasma membrane of the neuron. We know that plasma membrane means in this case it is very long cell, and if I take the axon is nothing but very long and cylindrical, and it has surrounded by two plasma membrane. This is the one plasma membrane and this is the other plasma membrane, and you could see this is the lumen of the axon, and this is filled with a liquid, and that is called as axoplasm. So simply we write that is called as axoplasm, a liquid. Okay, now this particular ion channels, potassium ion or sodium ion channels, actually transport sodium or potassium with the help of ATP. So it actually transport these molecules or ions with the involvement of ATP, that is called as active transport. They need ATP. Actually, if you look into the structure of sodium or potassium ion channel, they have active ATPase binding site. That because they have to utilize ATP for the transport of the molecule. So this is the one thing to note. So we'll just keep it. Now, the step number one, the first step is when the neuron is not even conducting any of the action potential. There is no stimulus here. We don't have any stimulus. We don't have it actually. Got it? So it is completely at rest. So I just how to tell you, polarized means the charge, which is, you could see here in the axoplasm, you could see negative charge. So we have negative charge. Why negative charge? I'll tell you. And outside the plasma membrane, that is in the extracellular fluid, that is outside the plasma membrane or outside the neuron, you could see positive charge, correct? So this is positive charge outside. Inside it is negative. Outside positive, inside negative. I'll tell you the reason behind it. The reason is when the neuron is not conducting action potential, it is actually at rest. It is completely at rest. And you could see the ions here. So here we have inside the axoplasm, we have 4 sodium ions, and we have 9 potassium ions. So when you compare these molecules, we have more number of potassium ions inside the axoplasm compared to the sodium ions, correct? So we have high concentration of potassium ions. At the same time, you could see a sky blue color small structures are inside. What are they? They are nothing but negatively charged protein particles. They are negatively charged proteins. Got it? So we have high concentration of negatively charged protein and high concentration of potassium ions and low concentration of sodium ions inside the axoplasm. But outside you see exactly opposite condition. That is, we have more number of sodium ions compared to the potassium ions. Here we have 8 potassium ions in one side of the plasma membrane, that is outside of the plasma membrane, compared to the potassium ions. We have 2. For 8 sodium ions, we have only 2 potassium ions. That means the concentration of sodium ions is always high outside when the neuron is not conducting any action potential. It's completely at rest. That is called as polarized condition. Here you see two types of concentration gradient, correct? Okay, what type of concentration gradient you see? If you take the chemicals, that is ions are nothing but chemicals here. Okay, so we have more number of potassium and here we have less number of potassium. So there is a concentration gradient which develops across the membrane. If I talk about potassium ions. If I talk about sodium ions, here we have less sodium ions, outside high concentration of sodium ions. So again, concentration gradient builds across the membrane. This is called as chemical potential difference. Chemical potential difference builds, number one, during polarized state. Okay, the difference could be seen between the ions. At the same time, we see the charge, that is nothing but electric potential difference also seen. That is, outside we have positive charge outside the plasma membrane, but inside, that is in the axoplasm, we have more negative charge. Got it? So this is actually electrical potential difference. So there are two types of potential difference builds within the plasma membrane of the neuron and outside, that is ECF of the neuron. So this is at polarized state when neuron is not conducting any action potential. Hope you got this point now. Now we'll go to the step number 2. That is actually when a stimulus is introduced here. Okay, let us assume that this is actually the tip of the neuron, and somewhere else the stimuli actually has arrived, or like it is present now. Okay, now it comes into contact with one site of the neuron. This is the site number 1, this is the site number 2, this is the site number 3 of the same neuron. So we have 3 sites here for our understanding. Okay, so we just write that this is site number 1. So let us just name it as A here, and this part is site number 2, that is B here, and this part is site number 3, that is C here. So we have 3 sites A, B, C. The stimulus is received by site A, not site B or not site C. They have to receive later. Okay, first part of the neuron which receives the stimulus is called as site A. Okay, so now when the stimulus is applied here, and now you will see the second step, that is actually action potential. That is nothing but now polarized state will be depolarized. Everything will be reversed. So what you see is during the depolarization, more number of sodium ions with the help of sodium ion channel through active transport, they now start coming inside the exoplasm. Okay, so like this, you could see here more number of sodium ions are getting inside. So now if you just count the sodium ions here, 1 2 3 4 5, we have 5 here, 2 3 potassium ions. So the concentration of sodium ion in the site A is increased when the stimulus arrives, okay, compared to the potassium ions. This will make the entire site A to reverse its charge. See, negative charge earlier, now it will become positive, and whereas the negative charge goes outside. So this is reversal of the charge. Again, you could see electrical potential difference, that is reverse. Okay, so again, you could see here both the sides of the plasma membrane, you could see the reversal of electrical potential difference. At the same time, you could see now sodium ion concentration is now reduced, very low here outside. We don't see any sodium ions here, but inside, because of the rapid influx of the sodium ions when the stimulus arrives, you could see more number of sodium ions inside compared to potassium ion. This is nothing but high concentration of sodium ions. So again, you could see chemical potential difference. Okay, so this will be seen. So this is very important. And now this entire site A is depolarized. That is nothing but called as action potential. That means action potential is generated here. Okay, because of the disturbance. So disturbance in the membrane, charge as well as chemicals. Okay, so this is called as action potential. Hope you got this now. Okay, now the action potential which is generated at site A now travels to site B. That means here what you could see is again there is rapid influx of sodium ions like this. So they again come inside like this. Okay, more number of sodium ions actually get inside the exoplasm of the site B. Okay, whereas let me just tell you here, whereas in the site A we need to regain this condition, the polarized condition here, because there is no action potential. We don't want action potential which was already generated and conducted to site B. Now we don't want, we want to restore this condition as polarized state. Okay, and the mechanism is called as repolarization. Got it? So repolarization is taking place at site A. Okay, so now during repolarization, 3 sodium ions which are inside the exoplasm now get transported to outside to the ECF. Okay, so you see here 3 sodium ions, and now 2 potassium ions get inside the exoplasm, okay, to balance the charge. Now you could see restoring of the charge also. Inside becomes negative, outside becomes positive. Okay, whereas here also same thing, the other side of the membrane. Correct. So everything is restored, that is back to normal. This is called as repolarization. Now this particular site A is polarized. It will wait for another stimulus to produce action potential, that is reversal of potassium and sodium ions, at the same time reversal of the charge also at site A. Whereas in the site B you see depolarized state, that is nothing but action potential is again produced here. How? You could see reversal of the charge, and here also same thing, reversal of the charge. That means again electrochemical potential difference builds across the plasma membrane. So this is how it works. Got it? So we have more number of sodium ions because of the influx of the sodium ions through this ATP or active transport sodium ion channel. That's it. Okay, so compared to potassium ions, so we have less sodium ions here. This is it. So this is actually all about action potential or nerve impulse, and the process will be repeated. So once action potential is generated and it will be conducted to site C of the same neuron, and again here repolarization takes place, that is getting back to normal. Here what you expect is 3 sodium ions again is transported to outside, that is efflux of the sodium ions. For 3 sodium ions we have, I don't have, yeah, here, okay, so we have to get back 2 potassium ions. Okay, so we have one more here. Okay, so through this, because they are selectively permeable. Got it? So this is actually the thing works. This is actually repolarization. Hope you understood this concept. I believe that this session was kind of informative for you. Thank you.